Hello, welcome to Biology, the scientific study of life. Combinatorial control of gene activation. In eukaryotes, the process control of transcription depends largely on the binding of activators to DNA control elements. Considering the very large number of genes that must be regulated in a typical animal or plant cell, the number of complementary different nucleotide sequences found in control elements is surprisingly small. A dozen or so short nucleotide sequences appear again and again in the control elements for different genes. On average, each enhancer is composed of about 10 control elements, each of which can bind only one or two specific transcription factors. The particular combination of control elements in an enhancer associated which a gene turns out to be more important than the presence of a single unique control element in regulating transcription of the gene. Even with only a dozen control element sequences available, a large number of combinations are possible. A particular combination of control elements will be able to activate transcription only when the appropriate activator proteins are present, such as at process time during development or in a particular cell type. The example shown here in this figure illustrates how the use of different combinations of control elements to activate transcription allows exquisite regulation of a transcription with a small set of control elements. Notice that here represents DNA, this line, notice that. And this one line, yellow one, represent distal control elements. Notice these are enhancer, and this one activator. Notice also theta box here, green one, and the bromotor, dark green. Notice that these are a bromotor and this one gene. Closer look to theta box, DNA binding protein and group. These are group of mediator proteins and small one, each one is general transcription factors. Notice that. RNA, closer look, RNA polymers second. Closer look, notice also transcription. Back to the stage. Notice also activator proteins bind to distal control element grouped as an enhancer in DNA. This enhancer has 3 binding sites. Notice also here DNA binding protein. A DNA binding protein brings the bound activators closer to the promoter. Other transcription factors, mediator protein and RNA polymers are nearby. Notice also here RNA polymers, RNA synthesis, and transcription initiation complex. These are, notice also this stage, the activators bind to certain general transcription factors and mediator proteins, helping them from an active transcription initiation complex on the promoter. Coordinately controlled genes. How does the eukaryotic cell deal with genes to related functions that need to be turned on or off at the same time? In chapter 18, we learned that in prokaryotes, such coordinately controlled genes are often clustered into an operon, which is regulated by a single promoter and transcribed into a single mRNA molecule. Thus, the genes are expressed together, and the encoded proteins are produced concurrently, with rare exceptions. operons that work in this way have not been found in eukaryotic cells. Recent studies of the genomes of several eukaryotic species have found that some co-expressed genes are clustered near one another on the same chromosome. Examples include certain genes in the testes of the fruit fly and muscle-related genes in a small worm called a nematode. Unlike genes in prokaryotic operons, however, each eukaryotic gene in these clusters has its own promoter and is individually transcribed. The coordinate regulation of clustered genes in eukaryotic cells is thought to involve changes in the chromatin structure that make the entire group of genes either available or unavailable for transcription. More commonly, co-expressed eukaryotic genes such as genes coding for the enzymes of a metabolic pathway are found scattered over different chromosomes. In these cases, coordinate gene expression seems to depend on association of a specific control element or combination of elements with every gene of a dispersed group. Copies of the activators that recognize these control elements bind to them, permitting simultaneous transcription of the genes no matter where they are in the genome. Coordinate control of dispersed genes in a eukaryotic cell often occurs in response to external chemical signals. A steroid hormone, for example, enters a cell and binds to a specific intracellular receptor protein, forming a hormone receptor complex that serves as a transcription activator. Notice these, and this one, Every gene whose transcription is stimulated by a particular steroid hormone, regardless of its chromosomal location, has a control element recognized by that hormone receptor complex. Many signal molecules such as non-steroid hormones and growth factors bind to receptors on a cell's surface and never actually enter the cell. These kinds of signal can control gene expression indirectly by triggering signal transduction pathways that lead to activation of particular transcription activators or repressors. The principle of coordinate regulation is the same as in the case of steroid hormones. Genes with the same control elements are activated by the same chemical signal systems. For coordinating gene regulation probably arose early in evolutionary history and evolved by the duplication and distribution of control elements within the genome. Back to this, notice that control elements, these are enhancer and promoter. Notice that notice also crystalline gene, and notice this one, lens cell nucleus and liver cell nucleus. Notice available activators here, and notice also gene expressed, not expressed. Notice also crystalline gene not expressed, and this one gene expressed. A represent liver cell and gene is expressed and the crystalline gene is not. Lens cell, the crystalline gene is expressed and the albumin gene is not. Notice also the cell type specific transcription. Both liver cells and lens cells have the genes for making the proteins albumin and crystalline, but only liver cells make albumin, a blood protein, which means a blood protein, and only, notice here, okay, this is important, lens cell make a crystalline, the main component of the lens of the eye. The specific transcription factors, activators, and repressors made in a particular type of cell determine which genes are expressed. In this example, the genes for blood protein, which mean and the crystalline, are shown at the top here. Notice that. Boom. each with an enhancer made up of 3 different control elements. Although the enhancer for the 2 genes share on control elements, each enhancer has a unique combination of elements. All the activators required for a high level expression of the blood cell gene are present only in liver cells A, whereas the activator needed for expression of the crystalline gene are present only in the cells B. For simplicity, we consider only the role of activators here, although the presence or absence of repressors may also influence transcription in certain cell types. Okay, thanks for